All right, so we're going to try and finish these notes here today. We left off on Golgi bodies, Golgi apparatus. Either term is fine, referred to both ways. So the ER, if we just think back, the ER is a series of membranes with ribosomes attached, okay, or some of it does not have ribosomes attached, where materials are produced and then moved from one place in the cell to another. Oftentimes, those materials then get sent to the Golgi apparatus, which a lot of times looks similar to the ER. It's also a series of membranes. However, it has a sort of different function. Okay? The Golgi apparatus takes materials that are going to be sent out of the cell and sort of packages them up. Okay? And the way it does that is it puts those materials into little bubbles we might call vesicles. It could modify the products that have been produced in the ER. And again, it has sort of two sides to it. Just like the ER had what's called the cis phase and the trans phase. Okay? The trans phase is the side that sends these vesicles out that are going to be leaving the cell. Okay? And so what would be happening is, like, the ER might be over here. It would send materials to the cis phase of the Golgi apparatus. Those materials could be rearranged, could be modified in some ways, eventually make their way to the trans phase, get put into these little bubbles, which then head out to the end of the cell membrane, merge with it, break open, and then release their products outside of the cell. So this is packaging things up that are leaving the cells. There are no ribosomes in the Golgi apparatus, so those ribosomes are in the ER. And so the Golgi apparatus receives those things. So again, you see the same kind of thing. Here we have the cis phase that is receiving material from the ER. It empties that material into the membrane. They make their way from membrane to membrane. Eventually, after they've been modified, they get sent out in these vesicles. So a cell that is producing a lot of products that are going to leave the cell, like cells that produce hormones, produce the hormone inside of the cell, then send it out through the Golgi apparatus to be released into the bloodstream where it can be shipped around the entire body. So secretory cells, cells that secrete a lot of products, have a lot of Golgi apparatus in them. The lysosome, do you remember what does the root lysis mean? Like hydrolysis? Breaking down. The lysosome is an organelle that contains enzymes that are hydrolytic enzymes, enzymes that are going to break down material. And they might do this for lots of different reasons. So sometimes cells like white blood cells have to destroy pathogenic cells, bacteria, or to destroy viruses or other pathogens. And so the lysosome can help to do that, can release these enzymes that destroy another cell. Also, if the cell needs to digest large molecules within it, the lysosomes can release those enzymes to help break down large molecules. They also are used for sort of recycling organelles that are no longer useful. Sometimes organelles are sort of have run their life, they're used up, they don't function anymore. The lysosome can sort of break them down and reuse their parts to make new organelles. So they just look like small little sacs within the cytoplasm. There's nothing really distinguishing about them. You can't see anything inside of them, but that's what their function is, to break down these materials. So here you see some examples. If this cell is, if food particles, large food particles, are being engulfed into this cell, what process is this called? We learned about it the other day. Pinocytosis, the indenting of the cell membrane to ingest the material. So this big chunk of food is now inside the cell. It might be called a food vacuole. A lysosome then can travel to that food vacuole, open up, release these enzymes into it that can then digest those larger molecules into smaller molecules. So that's what the lysosomes do. Vacuoles are storage areas. They're surrounded by a membrane, and there's several types of vacuoles. In plant cells, for example, there's a large central vacuole that fills up a large chunk of the interior of the cell, that exerts pressure on the cell wall, that gives the plant cell its support. In single-celled organisms, there are special vacuoles called contractile vacuoles. They pump excess water out of the organism. This is especially important in freshwater cells. Because what happens with cells that are in fresh water? Thinking back to osmosis and diffusion. Caitlin? They swell up. Because there's a higher concentration of water on the outside, water is constantly moving into them. So they need some way to get rid of it. The way they do that is through these contractile vacuoles, where that brings water through these arms into the central area, it squeezes and pumps that water out. This is a paramecium. Okay, here's a central vacuole in a plant cell. Food vacuoles are used to store food and allow it to be digested. The mitochondria.
Powerhouse of the cell.
The organelle in which cellular respiration takes place.
Can we put like common powerhouse?
No!
I'm writing
I already put
powerhouse
powerhouse
itself.
on there.
I'm going to make a multiple choice question on your next test. I'm going to make powerhouse an incorrect answer.
I'm gonna put
Take
powerhouse
that.
either way. I'll take
Everyone
that.
You'll take the point, you're going to take
Why do you have
it.
to take
We're all gonna get it wrong.
We all get it wrong. None of us get it wrong.
All right. So the mitochondria is where cellular respiration, we are going to spend quite a bit of time talking about the details of cellular respiration in the next part of our unit. But for now, we'll just go over sort of the 7th grade version. Cellular respiration takes in glucose and oxygen, breaks it down, transforms that energy into ATP, which the cell can use, and releases carbon dioxide. So mitochondria are sort of spherical shapes. They have a double membrane, an outer membrane, and then a very white, intensely folded membrane on the inside. And the folds are called cristae. And what they do is they allow there to be a much larger surface area. Having this winding membrane inside creates a large surface area because embedded in that membrane are proteins that allow the reaction to take place. They're enzymes. Okay. And mitochondria, they contain their own DNA, separate from the DNA that's in the nucleus. They also contain their own ribosomes. Why is that? Why would mitochondria have its own DNA? They can replicate on their own within the cell as well. Dean?
A really long time ago, we had our cells, and then the mitochondria was like bacteria, so they like merged.
Yeah. We learned about the evolution of the first eukaryotic cells, and chloroplasts and mitochondria are thought to have been prokaryotic cells that entered into eukaryotic cells and formed a permanent symbiotic relationship. So that's why they have their own DNA. Typically, a cell might contain a few hundred of them. However, cells that are very active, that require a lot of energy, might have more of them. They also, the number changes. It's one of the reasons we adapt to things like exercise, and we become more efficient at running or swimming or dancing or whatever you do that uses your muscles, is that your muscles, as you stress them and use them more and more, they start to develop more mitochondria, and therefore they can release more energy, allowing you to have better endurance at using those muscles. Here's what they really look like in an electron microscope image, and you see the outer membrane and then this folded membrane that's inside of that. Chloroplast Again, another organelle that has its own DNA, has its own ribosomes, also evolved from this symbiotic relationship with prokaryotes. It's where photosynthesis takes place in plants and in other autotrophic organisms. It's green. Why? Contains chlorophyll. Chlorophyll is a green pigment which absorbs photons of light and starts the process of photosynthesis happening. They also have a double membrane, outer membrane and inner membrane, and then the inside is called the stroma, this fluid-filled interior, which then has in it these, look like stacks of poker chips or macaroons or something. And they're called thylakoid, is the name of the individual poker chips, and then the stacks are called the brana. Here's what it looks like in the electron microscope. Again, sort of the shape, and you can see the stacks, the thylakoids inside of this chloroplast. Here's what they look like in the light microscope. Okay, you see these are obviously plant cells. We have the white area is the large central vacuole, and the chloroplasts are sort of pushed to the outside. So we saw this video, or this was in the video we saw before. And so we sometimes imagine cells to just be like a balloon filled with a liquid. But they actually have what's called the cytoskeleton that helps give them their structure. Cytoskeleton is this network of fibers that sort of crisscrosses the cell. It does a couple things. It's important for maintaining the shape of the cell in animal cells. Because animal cells have that flexible cell membrane, you might expect them just to like turn into a flattened puddle or something. But they don't. They can maintain their shape because crisscrossing them are these fibers that hold them together and give them a definite shape. But they also are important for the movement of materials within the cell. For example, these are vesicles. We talked about earlier with the Golgi apparatus. These vesicles leave the Golgi apparatus and are heading out towards the cell membrane. Well, this animation is sort of showing how that would happen. This is a microtubule. This end down here might be connected near the Golgi apparatus, and the end that's being walked toward might be towards the cell membrane. So this motor protein, this is a protein, okay, that sort of uses energy to transport this vesicle from one part of the cell to another. So that's sort of how these materials actually move. They're not swimming around in there. They're being moved from place to place by certain molecules. So cytoskeleton is important in the movement of parts and the movement of things inside the cell. So there's a few parts of this cytoskeleton. One part are these things called the centrioles. And the centrioles are most pronounced in animal cells. They look like two rigatoni. One is vertical and one is horizontal. They're found generally near the nucleus, and they're at right angles to each other. And they're made of these rings of microtubules, which we'll talk about. And they are helpful when a cell is dividing and forming the spindle, which pulls the chromosomes apart. Here's what they look like in the electron microscope image. Here's the one that's sort of laying down horizontally. And then here's the one you're looking into. It's arranged vertically. So microtubules are these small hollow cylinders. When we saw that motor protein, it was walking its way along the microtubule. So they're long, hollow cylinders that are found stretched across the cytoplasm. They're also important parts of the centrioles. They make up cilia and flagella, okay? And they serve as sort of a track where those motor proteins can walk the materials around. Microfilaments are also part of the cytoskeleton. They're thinner, however. They're just a solid strand, like a strand of thread. They also, these microfilaments, are able to contract. When they work together, they can contract, and so they're a main component of muscle tissue. Muscle tissue, which has the ability to contract, to move our bones, has a lot of this, these microfilaments. It's made of a material, a protein called actin. And they're another part of the cytoskeleton. So flagellum and cilia are found in some cells. They are also composed of microtubules. Flagella are used for locomotion, for the movement of a cell. Things like bacteria often have flagella they use for movement, but also cells and other organisms, multicellular organisms, have flagellum. Things like sperm cells have a flagellum, which allows them to swim to reach the egg. When we looked at the Protosublina, had a flagellum that pulls it through the water. And it's kind of interesting. So this is the flagellum, the tip of the flagellum. And within a cell, there's actually a cellular motor. This diagram here might look like a technical drawing you might see in like DDP, a tiny electrical motor. It's actually made of all organic material. It's proteins and things like that that are arranged that actually take in ATP and spin around, allowing the flagellum to actually move. So it's like a living motor within the cell. Cilia are similar. They have basically the same structure as a flagellum, okay? But they're smaller. Okay? They're also made of these microtubules. Okay? They attach through the cell membrane, and they are used also for locomotion, for movement in some organisms, like in paramecium. But also, they are used in multicellular organisms. We have cilia on some of our cells. Cells in our trachea have cilia to filter out little bits of contaminants in the air so it doesn't get into our lungs. The fallopian tubes, the ovary in females releases eggs. They're not physically connected, however, to the fallopian tube. They're nearby. And the fallopian tube has cilia on it that kind of move together in unison to sort of bring the egg and have it travel down the fallopian tube. The cell wall is the outer shell of many cells. It's rigid. It's non-living. It's sort of produced and then it stays that way. It's not flexible. It doesn't really regulate material coming in and out. It gives structure. It gives protection to the cell. It does have small openings in it, which allow for materials to pass through to get to the cell membrane, where they can then diffuse in or be transported in by proteins. And depending on what type of organism it is, the cell wall might be made of different materials. In plants, it's made of cellulose, polysaccharide. In fungi, it's made of another material called chitin. Bacteria, it's made of peptidoglycan. It just depends on the organism what determines what its cell wall is made of. Any questions about any of our cell organelles that we've covered? All right, so our plan here is we have a lab to work on over the next couple of classes. We also have another one of those worksheets about the actual cell organelles. We'll have a review set of questions, and we'll have a quiz probably end of next week, I would say. So then we'll move on to the next part of our unit when we're done with that.